If there's no questions, let's go ahead and get started. So if you look on the schedule, we're doing host defenses 2, and probably tomorrow I've split the host defenses 2 and I made a host defenses 3, and I put that up, and I think we'll get started with that tomorrow. I broke it in, I beefed it up a little bit and I broke it off, but I think we can get that done in one day. We might finish host defenses 2, but probably not today. Any questions? All right. If there's no questions, let me go ahead. I don't think I've started. I started recording it, but let me share the screen. Oops. Nobody mentioned that. I just showed you the schedule and I hadn't shown the screen. So we will work on host defenses 2 today, and we'll be close to finishing, I think. Let me mark that. And tomorrow we'll have a longer lesson, meaning both 1 and 2 will be on the lecture, and so we should finish up host defenses 2 and then start and finish host defenses 3. Any questions? If not, I don't think I need that. Let's turn to the lesson. There it is. When we were talking about host defenses 2, I was talking about the lymphocytes, and that were the B and the T lymphocytes, and we talked a little bit about that and mentioned that both of the lymphocytes start their life from the stem cell that is found in the adult bone marrow. So in adults, B and T cells start from the stem cell, which can be found in the marrow of our bones. Any question about any of that? If not, maybe I should put this at the top. Nope, it's not going to work there. Let's put it way over there. So we need to talk about the T cells, the immature T cells, which are in the bone marrow of the adult. They will migrate out of the bone marrow and move to the thymus, where they will mature, and they'll stay there until they become mature T cells, although you can question how mature they are, because they're not fully mature. But they mature in the thymus, and then they start moving around the circulation. Let me show you a picture of the thymus, if I can get it. Let's blow that up. Recall that the thymus is a gland really close to the heart, and that's where the T cells mature, and then they enter circulation. They can, after leaving the thymus, they can directly enter the lymphatic system, which is the veins and nodules shown in green here. And if you notice, the lymphatic system pretty much circulates around the body. It is missing a little bit in the abdominal cavity in certain places. Actually, that's the pleural cavity, sorry. And it's not found in the lungs, but it has vessels going throughout the body, particularly the breast, but into the brain and the muscles and essentially everywhere. It is a very complete circulatory system, not quite as complete as the circulatory system. And in fact, at this part here, the lymphatic duct, that's where the lymph system dumps into the circulatory system. So the two are connected. as well as at the various organs of the lymphatic system. What am I trying to say here? That the lymphatic system, it drains the plasma that leaks out of the circulatory system, particularly from the capillaries, and then the plasma bathes the tissue in fluids, and that plasma is connected or collected in the lymphatic system, and then that's sent through the vessels and then through the nodes where the fluid is checked for pathogens, and then it's eventually brought up to the lymphatic ducts where it's dumped into the circulatory system, and then the plasma rejoins the blood. Any question about any of that? Let's see if I can shut this down. Well, it looks like no, I didn't. I shut the wrong one down. Darn it. All right, let's get that. No, there it is there. Let me shut it down from here. So the T cells, they mature in the thymus and then leave the thymus and then circulate around in the body. While the T cells are in the thymus, the immature T cells, they divide rapidly and mature. And when they come out of the thymus, they have the ability to recognize foreign antigens. And the ones that recognize the foreign antigens survive. We'll talk a little bit more about that later. And let me remind you, an antigen is just a molecule on the outside of a cell that has infected the body, and the antigens are what the host responds to, meaning the molecule that the host responds to and recognizes as separate or an entity invading the body. And they are a molecule. They are a molecule of a foreign cell, but remember the host immune, even though it reacts to that antigen, it specifically reacts to only one part of that antigen, and those we call antigenic determinants or epitopes. And a T cell which reacts to an antigen reacts to one epitope, and so each T cell that comes out of the thymus reacts to one epitope. Any question about any of that? And in about 2 to 3 days, once the T cell is moved to the thymus, it matures and then leaves the thymus, and then circulates around in the body, generally entering first into the lymphoid or the lymph tissue, meaning the lymph fluid, but then can enter the blood and then can circulate into the tissues and the organs. Any question about any of that? All right. There are several types of T cells, and we say that the T cells are the cell-mediated arm of the immune system. It's called the cell-mediated arm of the immune system because this part of the immune system is all cellular, meaning everything is done by T cells, and although they can secrete chemicals, they come from the T cells. And these cell-mediated arm can do different things. Some of the T cells can find a cell that has the antigen that the T cell is responding to, and the T cell can bind to it and then actually lyse that cell. And we'll talk more about the T cells in a little bit after we talk about the B cells, but some of the T cells can actually lyse foreign cells that enter the body. And then the T cells, although they can do that, they are much better at recognizing bacteria and viruses that infect and get inside our own host cells. They can attack fungi, protozoa, and helminths, so they can do that. And they can attack bacteria cells that are freely floating in the blood, but they're not good at doing that. They're much better at finding the bacteria when they've infected our host cells. And we haven't talked much about that, but you know that viruses infect host cells and they replicate in the host cell. Well, some bacteria can do that too and get inside our host cells and infect them. And the non-cell mediated arm of the immune system isn't good at finding those bacteria that are within our host cells, but the T cells are good at it. And besides having T cells that go out and attack cells that are invading our body or viruses that infect our cells, there are other T cells which really help activate the other cells of the immune response. And these T cells we call helper T cells. And they can activate white blood cells, especially the B cells, but also macrophages, and help them to fight off an infection. Okay? Any questions about any of that? The cell-mediated arm of immunity is the arm of the immune system that is responsible for rejecting foreign tissue, like in a tissue transplant or an organ transplant. And these patients, because the immune system will eventually respond to that tissue transplant, unless you get it from an identical clone, meaning you get it from an identical twin, your immune system will reject that tissue transplant. And so patients who get a tissue transplant, they have to go on immunosuppressive drugs. And that's to make sure that the T cells don't reject the tissue transplant. Because no matter how good the match is, unless it's coming from an identical twin, the T cells will eventually realize that that is a tissue transplant and then will mount an immune response against it. And that's why when you get a tissue transplant, you always have to take immunosuppressive drugs. Usually the immunosuppressive drugs only need to weaken the immune system a little bit. They don't need to remove the immune system. They just need to weaken the immune system a little bit. Excuse me here, I'm having a little bit of trouble today. The cell-mediated arm of the immune system, meaning the T cells, are also an important factor in the immune response against cancer. So if a patient gets cancer, and if their immune system is reacting and attacking that cancer, it's because of the T cells. Any question about any of that? All right, I've got a little picture here about B cells, and I'll talk about that in a minute. So let's move on and talk about B cells briefly. B cells also start life from the stem cell in the adult in the bone marrow. The difference is, unlike the T cells, the B cells stay in the bone marrow until they mature. And once they mature, they then migrate to the lymphoid tissue, meaning the lymphatic system, or the blood, meaning the circulatory system. And then they circulate around in the body in the lymph system and in the circulatory system. Any question about that? Well, once B cells mature, they can recognize antigens, and each B cell on their surface has a B cell receptor, or BCR for B cell receptor. Let me see if I got that. Yes, I do, right here. So these are the B cell receptors on the B cell, and the B cell can have 500,000 copies of the B cell receptor, but each B cell only expresses one B cell receptor, 500,000 copies of that one B cell receptor, so that each B cell receptor on a B cell can only bind to one epitope of one antigen. Is that clear? So that means each B cell can only bind to one thing. How in the world does our immune system respond to all the different antigens and the vaccines we ever take if the B cells receptors on the B cell only respond to one antigen, actually one epitope of one antigen? All right, who can answer that? Our immune system responds to a lot of different things. I'm sure you've each gotten different vaccines, and you've mounted immune response to that vaccine, and that's why you're protected for whatever the vaccine was trying to protect you from. And if each B cell receptor on a B cell can only bind to one thing, and that's 500,000 copies on the B cell, but it only binds to one thing, how come our immune system responds to so many different things? You guys are not answering today. JC, you have an answer? Ian, you have an answer?
Is it broad what it can bind to, or is it specific?
No, the B cell receptor can only bind to one epitope of one antigen. Is that your question? So it can't bind to anything else. It can only bind to one thing. Okay, Layla, do you have an answer? Yolanda, Anthony. Boy, we have a short class today. Nobody has an answer. Should we just sit here and wait? The reason why your immune system can respond to all these different things that it's challenged with is because you have 1 to the 10 to the 9th B cells. That is, let me think, is that a billion B cells? Sorry, I'm losing my memory here. Yeah, it's a billion B cells. And each of them can bind to a different thing, meaning that you can bind to 1 times 10 to the 9th different things. And that doesn't mean that you can respond to every antigen or respond to everything, but it means you're essentially capable of responding to almost all antigens. And so your immune system can respond to almost everything. Any question about that? So when your body is attacked by a pathogen, your immune response will respond to it, and the reason why is you have 1 times 10 to the 9th B cells, and the chances of at least one of those B cells responding to an antigen on that pathogen are really good. Any question about that? Let's open this in a new tab, and let's watch a little video on how B cells, once they get stimulated, they go through clonal expansion. There it goes. This might be a little small. See if I can blow that up. Nope.
Your blood contains a wide variety of B cells.
Is that loud enough for you guys to hear?
Yes.
I'll try and turn that up a little.
Your blood contains a wide variety of B cells, each of which bears a unique type of antibody on its surface. When your body is attacked by a foreign pathogen, antigens located on the surface of the pathogen will bind to any B cell with an antibody that has a complementary shape. The binding of the antigen to the antibody of a B cell leads to its activation. Once activated, the B cell will multiply rapidly to produce an army of genetically identical B cell clones. Following division, these clones will differentiate to produce cells that will mount a direct attack on the foreign invader. The clones will divide into one of two types of cells, the plasma cell or the memory cell. Plasma cells become antibody-producing factories. Memory cells hang around after the infection is over and play a role in future immunity to the antigen that stimulated their production. This graph illustrates how the production of antibody changes if you're exposed to the same antigen again. The level of antibody rises after the first exposure to antigen, then falls when the infection is defeated. Memory cells produced at this time can remain for months to years. Upon second exposure, these memory cells can immediately activate and begin producing antibodies. Because of the small army of memory cells waiting to be activated, the second response is both faster and larger than the first, so much so that the pathogen doesn't have a chance to cause illness.
Okay, any questions about this video? Come back to here. So the B cells, each of them have a different B cell receptor, which they hinted at what that was there, but we'll talk more about that later. And so when an antigen comes into the body, and usually a pathogen that's attacking the body has more than one antigen, but we're only going to look at this one antigen, and that antigen binds to only one of the B cells, because we're really talking about one epitope of one antigen, of one pathogen. And obviously a pathogen can have many epitopes, but we're going to ignore that and just talk about this one. And when this B cell gets stimulated, meaning that the antigen is binding to the B cell receptors, that activates that B cell, and then that B cell goes through clonal expansion, making an army of B cells that all react to that one specific antigen that first stimulated that B cell. Any question about that? And some of these cells mature into the plasma cell, and the goal of the plasma cell are to make antibodies, and then the plasma cell releases these antibodies, which go into the circulation, either in the circulatory system or the lymphatic system. And these antibodies move through the body to find and bind to the antigen and then attack that pathogen. Any question about that? We'll talk about later that some of these antibodies can even leave circulation and enter into the tissue. We'll talk about that in a little bit. The first time you're exposed to an antibody, your immune system, the B cells get activated, and then they'll respond and make a bunch of antibodies, and that'll defeat the pathogen, so it'll go away. And after the pathogen has gone away, there's no more stimulation of the B cells, and so they'll stop making antibodies, and then the antibodies will decrease. And eventually, at some point later, you won't have antibodies to that antigen circulating in your body. But there are memory cells, and where was that picture of that? Someplace after this. There are some B cells that differentiate into a memory cell, and the point of them is they're long-lived, and at some future time the pathogen infects the host again. There's these memory cells of the B cells, and they will respond to that pathogen and then go through a second clonal expansion and make antibodies. And this immune response will be quicker because the memory cells already exist, and they will respond to that antigen, divide, and then start producing antibodies. And as you can see, that response is much greater the second time than the first time, meaning we get many more antibodies produced. And that keeps the pathogen from causing disease the second time. And this is the entire point of a vaccine. The difference is the patient is given the vaccine, and the first time the patient responds, producing antibodies as well as T cells. We'll talk about the T cell part later. And it's a weak first response compared to the second time. But then there's memory cells, memory B cells and memory T cells. And then when the pathogen does invade the body, the immune response mounts a very great immune response. And that's why vaccines work, and why vaccines protect us from the disease that the vaccine is trying to prevent. And that is the disease doesn't get established in us because we get this second immune response. Any question about any of that? Let me move this over. Not quite. I can shut that down, come back here. So when the B cells first get activated, and that happens by this first B cell binding to an antigen, then going through clonal expansion, most of the B cells differentiate into plasma cells, and their goal is to produce antibodies and then secrete them. So the antibodies go into circulation to find or bind to the antigen and then attack those antigens. And that's called clonal expansion, and then differentiation into the plasma cell. But a few of those B cells differentiate into memory cells, and they're long-lived, and they're just waiting around to see if that pathogen sometime in the future invades the body again. And then they will respond the second time. The antibodies from the plasma cells circulate in the body's fluids, and by that it's the blood and the plasma, the lymphatics, the lymph fluid, and the mucus. We'll talk a little bit more about that in just a minute. And for that reason, the B cells are called the humoral arm of the immune system. Humoral means fluids, and it's called the fluid arm of the immune system because both the B cells and then the antibodies circulate in the body's fluids. And the antibodies can get into the blood, the plasma of the tissue, or the lymph fluid, as well as the mucus. And the antibodies can also go, besides in the mucus membrane, so I'm talking about the mucus membrane, the antibodies can be secreted into the mucus. But the antibodies are also secreted into mother's milk. Okay? Any questions about that? And so the antibodies are found in the fluids, and that's why we call it the humoral arm of the immune system, as opposed to the cellular arm of the immune system. The B cells make antibodies which are good at binding to antigens which are freely circulating in the body, like in the body's fluids, like a bacteria that's invaded the blood or a virus that's freely circulating in the blood. The antibodies aren't as good at reacting to a pathogen that has invaded our host cells. They can do it, but they're not as good at finding pathogens that are inside our host cells. They're much better at finding pathogens that are freely circulating in the body's fluids, like a virus that hasn't infected a host cell yet. If it's just freely circulating in the blood, antibodies can bind to it. And then bacteria that's freely circulating in the blood or the lymphatics. Any question about any of that? All right, so the antibodies bind to an antigen, and the main purpose of the antibody binding to an antigen is that when it does, it just flags this cell for a phagocyte and tracks phagocytes to this cell that the antibody binds to, as well as aids the phagocyte from engulfing or phagocytizing the pathogen. So that's the main purpose of the antibody binding to an antigen, just flagging that antigen for destruction by the phagocytes and then removing it from the blood. The antibody usually by itself does not damage the pathogen. Okay? Usually it just flags it. But there are several things that can happen, and these we call the results of what happens when an antibody binds to a antigen. The first thing you can have is neutralization, especially toxins, but also viruses can be neutralized when the antibody binds to the toxin. When the antibody binds to the toxin or the virus, that binding can change the protein folding pattern of the toxin or the virus, making the toxin or the virus non-functional. And then also neutralization can coat the toxin or the virus, making it, or even a bacteria cell can coat the bacteria cell or the virus or the toxin, making it impossible for the pathogen to bind to a human cell, meaning all of these pathogens have to bind to a human cell, and that's their host cell, and then when they bind, they can then enter that cell. And if you prevent the pathogen from binding to a host cell, it can infect the host cell, and therefore it doesn't do damage to the host cell. And that's neutralization. An antibody binding to a pathogen can also serve as opsonization. Remember we talked about that in our last session, that an antibody can bind to a pathogen and then flag the pathogen for the phagocyte to phagocytize this complex, meaning the antibody bind to the pathogen. So it's flagging the pathogen, but also the antibody acts as a bridge to help bind the white blood cell, the phagocyte, to the pathogen, meaning the antibody, one side of the antibody binds to the pathogen, and that's the Y side, and then the other side, I guess the tip of the Y side, the other side of the antibody binds to the phagocyte. And so this antibody is acting as a bridge to connect the phagocyte to the pathogen. And that's what opsonization is. Signaling the phagocyte to come over here, bind to this pathogen, and then phagocytize it. And we did talk about C3b serving as an opsonin. And I think I mentioned that antibodies can also act as an opsonin. Any question about that? The other purpose of an antibody binding to a pathogen is that you can cause agglutination. It's sometimes called also aggregation. If it's involving cells, they use the term agglutination. And then if it's non-cellular things, they use the term aggregation. And I'm just going to use them as synonymous because I'm not going to separate it out. The whole idea of the antibody binding to a pathogen is that each antibody can bind twice on this edge of the Y and at this edge of the Y, meaning that an antibody can bind to two pathogens that invade the host. And then when you have more than one antibody binding, it binds to other pathogens, and so we get an agglutination, a clumping of antibodies and pathogens. The advantage of agglutination is, well, first, instead of having 6 different regions of the body infected by the pathogen, you're only going to have one region of the body infected by the pathogen, meaning the pathogen isn't free to circulate. It's going to circulate only as this large clump. Is that clear? Instead of in 6 different areas, it'll be only in one area. This clump can be big enough that it'll actually precipitate out of the blood. And if it precipitates out of the blood, the kidneys will then remove it from circulation in the blood. And although the kidney has to deal with the pathogen, it's much better for the kidney dealing with this pathogen than having the pathogen freely circulating around the body in the blood. And then the other thing that agglutination does is it makes it much easier for the phagocyte to phagocytize the pathogen. Instead of the phagocyte having to move to 6 different locations in the body, it just starts phagocytizing this cell, and then it moves on to this cell, and then it moves on to that 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 cell. So instead of the phagocyte having to look for all 6 different locations in the body, it just has to start at one end of the clump and then work its way to the other end of the clump. And so agglutination aids the phagocytes so that they can do their job of phagocytizing the pathogen. All right, any other questions about agglutination and how it aids the body in defending the body? The next one I don't have a picture to exactly, although this can sort of show it. And that is that when the antibody binds to the antigen, let's show it here. The antibody binds to the antigen, well then the complement protein, C1, can bind to this antibody. And then that can activate the complement cascade so that a hole gets put in the membrane of this cell. So antibodies can aid the complement activation. And we talked about that last time. Activating the complement proteins, which then leads to the complement cascade, putting a hole in the pathogen. So the antibody itself doesn't lyse the pathogen, but it can work with the complement proteins to lyse the pathogen. Any question about that? All right. The next thing that antibody binding to a pathogen can do is that when the antibody binds to the pathogen, it causes inflammation in this region. So when an antibody binds to a pathogen, we get inflammation in this region, and that will speed up the arrival of white blood cells as well as bring other inflammatory chemicals to the area to attack the pathogen. The antibodies binding to the phagocytes can have, like the complement proteins, activate in that region, and some of the complement proteins cause inflammation, and the purpose of inflammation is to spot that area for defense and bring in both the inflammatory chemicals, the white blood cells to attack the pathogen, and then begin repair in that region. So that's the purpose of inflammation, and we talked about that last time. and host defense is 1. All right. The last result that I'm going to talk about is antibody-dependent cell-mediated cytotoxicity, where the antibody binds to the pathogen and then can bind to a phagocyte. But in this case, the phagocyte doesn't phagocytize the pathogen. What the white blood cell does is and it could be any non-specific immune cell binding to the antibody bound to the pathogen. What it does is it causes that white blood cell to come up to the pathogen and then non-specifically attack the pathogen by secreting toxic substances against the pathogen, which will lyse the cells of the pathogen. And we mentioned that natural killer cells last time, as well as eosinophils when they're targeting a helminth, that's the method that they use. They will come up to the pathogen and then secrete toxic substances on the pathogen, which will kill the cell. And the antibodies binding to the antigens on the pathogen can aid those white blood cells, which could be an eosinophil or a T cell or a NK cell, to bind to the pathogen and then secrete substances extracellular which will attack this pathogen. And that is called antibody-dependent cell-mediated cytotoxicity. Any question about any of that? The different ways that antibody binding to an antigen brings an immune response against that antigen. All right, so let's talk a little bit more about the antibodies. Let me get that out of the way. Put it on the bottom here. Antibodies are shaped like this. It kind of looks like a Y, but this can bend right here, and so it can form a T-like shape, meaning this can bend down, and that can bend down. So it is bendable or flexible, the antibody, and it can bend tighter Y as well. Each antibody molecule can bind 2 times, so that can bind at this site and it can bind at this site. And please recognize that this is the same epitope. So when this antibody binds to an antigen, it's only binding to one epitope of that antigen. And this antibody can bind to the same epitope. And that's why we can get agglutination. This region of the antibody varies between different antibodies, so we could have this same class of antibodies. I'll talk a little bit more what a class is, but the point is that if there are different antibodies, they'll bind to different epitopes. That's different from the antibody which is made from the B cell. It will only bind to each antibody that's produced from that B cell will only bind to the same epitope. Did I confuse anyone? Okay, so this portion that's a little bit darker from the rest of the molecule is called the variable region, because the amino acid sequence of the proteins of this antibody differ in this region depending on what epitope the antibody binds to. And they will have a different amino acid sequence in the variable region of the antibody if the antibody binds to a different epitope. And that's why it's called the variable region. It's also called the fragment that is antigen binding, or Fab. Where do I have that? There it is. Fab, because it's the fragment of the antibody that binds to the antigen. and they just call it FAB for fragment antigen binding for short. Okay? The other region of the antibody, this region here and this region here, is the constant region of the antibody. So all antibodies of this class will have the same constant region, and this is called FC region for fragment constant. By what I mean by same antibody class, if you have different B cells but they're both producing the same IgG, maybe you know that. If you don't worry, we'll talk about it. They're different antibodies making the same class of antibody. They will have the same FC region, meaning the amino acid sequence will be the same in all of the antibodies of that class of antibodies. Is that clear? And that's why this is called the constant fragment, or FC region, of the antibody. This region of the antibody binds to antigens. This region of the antibody binds to the white blood cells, the phagocytes and other white blood cells, as well as this is the region of the antibodies which bind to the complement proteins. Okay? Any question about any of that? Alright, so let's move on to the different classes of antibodies. There are 1, 2, 3, 4, 5. That's right, 5 classes. There it is, there, I didn't see that. 5 classes of antibodies, and they have slightly different shapes. The class of antibody means that if you have an IgG antibody circulating in your blood, and even though the different IgG antibodies bind to different epitopes here, they will all have the same